Welcome back, students. We will just continue with the life cycle of Fasciola hepatica. As we have discussed in the previous video lecture, the encysted Meracidium gets converted into the active Meracidium larvae, and this active Meracidium larvae gets converted into the sporocyst larvae, that sporocyst larvae into the radial larvae. So this is the detailed structure of the egg capsule. Then this is a Meracidium larvae. This is the sporocyst and the radial larvae, and this radial larvae, from this radial larvae, the Saccharia larvae emerges out through this birth pore. So let's pass on to this Saccharia larvae. This Saccharia larvae is about 6 millimeter long. Saccharia larvae, it's about 6 millimeter long, and it is oval in shape. As you can see here, it is oval in shape with a tail. So we can say that this Saccharia larva, it is a tadpole like larvae, and it is covered with a thick spiny cuticle below with muscles with the muscles and the mesenchyme cells. So the body of this Saccharia larvae, it is covered with a spiny cuticle below which there is the muscles and the mesenchyme cells, and it has two suckers, one on the anterior region, that is the oral sucker, and on the ventral side you can see a ventral sucker or the acetabulum. Then the alimentary canal, it is very simple. It consists of mouth. At the center of the oral sucker there is an opening called as a mouth, which leads into the muscular pharynx, which is followed by the esophagus and a U-shaped. This is a U-shaped gut cecum is present. U-shaped or the inverted U-shaped intestine is present, and here you can see the numerous flame cells are present, which is located inside the body. These are the numerous flame cells. You can see the numerous flame cells which is close to the inverted U intestine. You can see the numerous flame cells. So once again, this Saccharia, it has an oval body and a tail region. Then this suckers are present, intestine is present, flame cells are present. Then excretory tubules of the two sides open into the excretory vesicles. So this is a excretory tubule, opens into the excretory vesicles, which continues as a excretory duct. So from each flame cells, the small ducts arises that joins together to form a duct, and it reaches the excretory vestibule. From the ducts from both the sides joins together and reaches the excretory vestibule, and it further runs towards the posterior, and it arises the excretory duct, and it runs to the tail, and it again bifurcates and open outside by the nephridiopore. This opening is called as a nephridiopause. The body wall contains many unicellular cystogenous glands are present. So here many cystogenous glands are present inside the body, which secrete a cyst in the later development, and the body cavity is filled with the groups of germ cells. These are the germ cells. These are the germ cells which is present inside the Saccharia, and the Saccharia migrate to the pulmonary sac of the snail and finally escapes into the surrounding water. And unlike the radia, this Saccharia larvae does not reproduce. So you can't see any the smaller Saccarias which is present inside the Saccharia larvae. Unlike the radial larvae and the sporocyst, you can see the miniature form of the radial larvae inside the sporocyst, and also inside the radia you can see the radial larvae, and no such structures are seen inside the Metasecaria larvae. Then, unlike the radia, the Sacaria larvae does not reproduce, and it swims. So since it contains a tail region, this Sacaria larvae swims by lashing this tail and settle down on some water plants, and it loses a tail region. It loses its tail region, and it gets converted into the Metasecaria larvae. Okay, and it loses a so once again it loses a tail region and gets converted into the Metasecaria larvae by encysting the cyst. This Sacaria itself, by losing the tail, gets encysted by the secretion of the cystogenous glands, and the next larval stage is called as a Metasecaria larvae. Now let's see the Metasecaria larvae in detail. Structurally, this encysted Metasecaria larvae resembles a Sacaria larvae. The major difference is that the absence of tail in Metasecaria. The tail is absent in Metasecaria, and cystogenous glands, the so-called cystogenous glands, are absent in this Metasecaria. Okay, or we can say that they get degenerates in Metasecaria larvae. The flame cells are also present in this Metasecaria. It has a mouth that leads into the pharynx. There is an inverted U intestine in the body cavity. The germ cells are also present. Then it is able to remain viable for several years, but leads a passive life, and it can resist only the short period of drying. So this Metasecaria, it is encircled inside the cyst. The suckers are present. All the structures which is present in the Sacaria is present in Metasecaria. The only difference is that the tail region is absent. Cystogenous glands are also absent, and surrounding the body of the Metasecaria there is a cyst wall. There is a cyst wall in Metasecaria. So that is the difference between the Sacaria and Metasecaria. And this Metasecaria leads a passive life. Then this Metasecaria develops further only when this Metasecaria is swallowed by the sheep. So inside the body of a sheep, what happens is to this Metasecaria, the cyst wall is digested by the digestive enzyme. So in the digestive tract of the sheep, the cyst wall is digested by the enzyme of the host. The liberated young fluke penetrate the intestinal wall and gets into the body cavity to reach the liver and the bile duct within a period of 3 days after the injection by the host. Then what happens is that it settles down on the wall of the bile duct, and with the help of the oral and this ventral sucker, this Metasecaria gets attached to the wall of the bile duct, and it feeds on the bile duct epithelium, bile and the blood, and grows into the sexually mature adult in about the 12 weeks. So within 12 weeks, this Metasecaria, this Metasecaria gets converted into the adult fluke. Here it repeats a life cycle by laying the eggs. So again this life cycle gets repeated. So in the life cycle of Fasciola hepatica, the radia and the Sacaria multiply the number. This radia and Sacaria multiply the number parthenogenetically, which alternates with the sexual reproduction. And this type of alternation of generation is called as a allogenesis or heterogamy. It's called as a heterogamy. It's called as a heterogamy or allogenesis. So in the life history of the Fasciola hepatica, this radial larvae and the Saccharia larvae multiply their number parthenogenetically, which alternates with the sexual reproduction, and this type of alteration of generation is called as a heterogamy or allogenesis. And in Fasciola, all the larval stages are delivered from a single zygote, or all the larval stages arises from a single zygote, and this is known as a polyembryony. This is called as a polyembryony. It's called as a polyembryony. And the liver fluke also illustrates the pedogenesis, which means that the production by the larva forms before becoming the adult form. So pedogenesis is also taking place, that is, which means a reproduction by the larval form. So this larval forms, you can see this, from this porosis the radial larvae arises, from this radial larvae the Saccharia larvae arises. So before attaining the adult stage, the larva begins to reproduce, and that condition is called as a pedogenesis. For example, the radial larvae is capable of producing the daughter radia parthenogenetically, and this process is called as a pedogenesis. This Fasciola hepatica causes the liver rot in sheep by upsetting the normal functions of the liver, and the infections reduces the milk and the meat production, and it causes a great damage to the livestock. The breeding also substantially reduces. The death due to the infection is also common, and the liver fluke, which is a parasite in man, also causes the liver diseases and anemia. The certain control measures includes a therapeutic treatment of the infected cases and also destruction of this host snail. We can just destroy this host snail, and we can just control the infection of this Fasciola larval stage. The human infection can be prevented by washing, adequate cooking of vegetables before eating. So these are the control measures that we can just follow, and for the summary we can just say that the Fasciola, it is a digentic parasite, that is, it has a primary host and it has a secondary host, that is a snail. It causes a liver root disease. They have an indirect mode of development with many larval stages. You can see these are the larval stages, and Metasecaria, which is a free swimming larvae which penetrate into the body of a snail. After 2 weeks, the Metasecaria larvae gets converted into the radial larvae, and the radial larvae gets converted into the Saccharia larvae, then develops into the Saccharia larvae, which is a tadpole-like, and after 2 to 3 days, this free swimming larvae turns into Metasecaria larvae, and by getting encysted and becomes active only when it enters into the body of a only if this Metasecaria larvae enters into the body of a sheep. Then heterogamy, pedogenesis and polyembryony are common in this Fasciola. So that's all about the life cycle of Fasciola hepatica. This life cycle of Fasciola hepatica, you have to draw and study all these pictures, all these larval stages, and also this cycle you have to draw and study. And each larval stage can be asked for 2 marks question and also a general essay question. Explain the life cycle of Fasciola hepatica. And that's all in this video lecture.